In the previous video, we talked about homogeneous and heterogeneous and how we were able to make different substances and turn them into a mixture, whether they were a solution or not. In this video, we're going to be doing the quite opposite. We're going to be exploring the fields of chromatography. Chromatography is the process where you separate the substances in a mixture. So to show the process of chromatography, we're going to be using the following materials to efficiently show it. We're going to be having water in 3 small cups, coffee filters, 2 washable markers, and 1 permanent marker. On each coffee filter, we are going to be drawing a circle in the middle. Doesn't have to be perfect. Let's also draw a thick circle. It doesn't really matter what color you're using as long as the permanent marker and one of the washable markers are the same color and one of the other washable markers are a different color. Make sure you use the secondary colors, not the primary colors. And if you're wondering, the primary colors are red, blue, and yellow. So the secondary colors that you should be using are green, orange, and purple, or yellow and black. All right, I'm done thickening with this. Now we'll move on to the permanent marker. All right, we're gonna make this thick. Stick, all right. Done with that one, then we'll move on to the last one. Here we use brown. All right, now that we've done that, now we'll fold our coffee filter into a cone shape. And the way you can easily do that is by folding it in half and folding this part in half. Then after that, folding this part in half and and this part in half again. And we have it. And then after we'll put it in water, but we should make sure the water level is below the line. All right, now we will move on to the permanent marker. All right, there we are, permanent. Now lastly, we will do the washable marker. And again, make sure the water's below the line, okay? All right, so already from completing, you can see that right here, the color from the line is already spreading out towards the end of the sheet. The water is actually spreading the color. Meanwhile, it's working for the washable ones, but for the permanent, it's just staying there. We will wait for 5 to 7 minutes, come back, and let it dry. All right, we waited for about 5 to 7 minutes, and we're now gonna let this dry. So we'll start the first one. I'm gonna unfold it. So I see that the color that was originally in the middle has now been spread around the whole coffee filter. Keep like this over here. Then we're gonna take this next one. The water wet the whole thing, but what happened is that here with the permanent marker, I can see that it did not spread at all. And it's the same story with this one and this one, the 2 washable markers. The water helped the marker in the middle spread. We will let it dry, and then after that, we will analyze the colors. So it's been 30 minutes, and these are our dried coffee filters. So we can see over here that in this washable marker, we used green color, and we got results of blue and yellow color also appearing out. Here with the permanent marker, it stayed, not spreading out like the washable markers, And for this other brown washable markers, we have a combination of brown, purple over here, and orange. The reason why the permanent marker was not able to spread like these washable markers was because permanent marker is not soluble in water, unlike the washable markers, which are. So this concludes that chromatography is the separation of different substances in a mixture. The mixture here would be the washable marker would be the mixture containing different colors, and here it was separated. Scientists use it to find out viruses and medicines from complicated sources.